Dear students, now we are going to discuss inhibitors of enzymes and particularly importance of competitive inhibitors in medicine. What about the importance of competitive inhibitors in medicine? So first one, competitive inhibitors in medicine. Okay, so they are used in controlling diseases caused by pathogens. Okay, so they are used in controlling diseases by bacteria, for example. So how to control bacterial diseases or fungal diseases or some other microbial disease? For example, this is a host cell, human being cell, for example. Host cell. And inside that host cell, for example only, this is a bacterial cell, for example only. The bacterium. And this is a genophore, genetic material of that bacteria. And that codes for synthesis of an enzyme, whatever the type of enzyme. So this is a enzyme. This enzyme is able to convert a substrate, a type of substrate into some product. That product is a toxin. And that toxin is able to cause a disease. So because of toxin, the host is suffering from some disease. So how to control this disease? So here, the substrate concerned with the synthesis of toxin is A. Now, as your doctor, introduce or inject a chemical that is morphologically similar to A. For example, this is a A. This A looks like that A, but different. For example, blue in color and red in color. Chemically different, but morphologically similar. So same morphology. Morphologically, these two are similar. Morphology. Here, concentration of this one is only one molecule. But how many injected? For example, only 100 A type of chemical molecules injected into bacterial cell. So now the red colored A is competitive inhibitor of blue colored A. Now in this competition, instead of A number is very less. And what about the A number of this one? We injected is more. Now it enters into bacterial cell. So the A molecule which we injected is more. Now it is binded to the binding site of that enzyme. So once it is A, red colored one is binded to enzyme of that bacterial cell, this is enzyme, there will be no formation of toxin. No toxin, no disease. So A is morphologically similar to A present in the bacterial cell. A is specific. And the dummy one we prepared is red colored one. Its number is more. So simply, this is able to occupy the binding site of this enzyme. Actually, that enzyme is specific for this substrate. Once our chemical is inserted into binding site, there is no formation of concerned toxin, no disease. That bacteria will die because of lack of a concerned chemical. So definitely, we can cure certain number of bacterial diseases by competitive inhibitors. And second one, non-competitive inhibitors. Second one, non-competitive. So in non-competitive, inhibitors are morphologically not similar to the substrate. For example, this is a enzyme molecule, dummy one only. Sometimes you may take a globular protein. Shape of the enzyme is spherical. You can take like that also. Globular protein means round shaped protein. So this is an enzyme. And what about the substrate? So this is a morphology of a specific substrate. This is the real substrate. And what about the inhibitor? This is inhibitor. Morphologically, I means inhibitor. Inhibitor compound. So morphologically, these two are different. Okay, so now specific substrate is binded at the binding site of that enzyme. Gives rise enzyme substrate complex. So in this case, what about the attachment of substrate at the binding site? Okay, that we need. But what about inhibitor? Inhibitor is binded to the enzyme at location other than binding site. So inhibitor is binded at a site other than binding site. Okay, so now this inhibitor is able to change the morphology of the enzyme. So what about the enzyme? Enzyme morphology is completely changed. This is the enzyme. Okay, so what about the binding site? Widened. Once the morphology of enzyme is changed or binding site is changed, what about the substrate? Is it in contact with binding site? No. So here permanently, enzyme is permanently damaged. So in this case, both are means substrate and inhibitor both are in contact with the enzyme. Within the active site, specific substrate. Other than binding site, inhibitor. Now inhibitor is making the enzyme not suitable for biochemical reactions. So in this case, enzyme is permanently damaged. Okay, even if the concentration of specific substrate is increased, definitely 100%, there is no coming back. Means that enzyme is not able to gain its catalytic activity. So in this case, enzyme is permanently damaged. Enzyme permanently damaged. Unlike a competitive inhibitor, enzyme is permanently damaged. Okay, so competitive inhibitors, what are the examples? Heavy metals, copper, silver, cadmium, and other molecules. Heavy metals are the non-competitive inhibitors. They are damaging the enzyme permanently, rendering the enzyme not suitable for biochemical reaction. So enzyme is permanently damaged. Okay, so third one, feedback inhibition. Feedback inhibition. Feedback inhibition. Okay, so for example, A is the substrate, changed into, transformed into B product. B changed into C product. C changed into D product. For example, here I'm taking A to B, C, D, and take E also. Certain number. This is a dummy one. So initially A number is more. Initially A number is more. After certain period of time, what about the number of products? E products. 10 products are there. So in this metabolic pathway, what are the enzymes involved? Dummy one, enzyme 1, enzyme 2, enzyme 3, enzyme 4. So with the help of enzyme 1, 2, 3, 4, A is one by one gradually converted into B product, C product, D product, E product. After certain period of time, what about the number of E products? 10. What about the number of A reactants? One molecule only. So end product is accumulated. So accumulation of end product. Accumulation of end product. Okay, so once end product is accumulated, now end product is reacting with the first enzyme. So now E is competing for binding site of enzyme concerned with a substrate. Less number of A, more number of E. So end product is binded with E1. So there is no conversion of A into B. So feedback inhibition is a end product regulated by end product inhibition. So end product is able to inhibit, in this case, first enzyme. So this is a feedback inhibition. So number of kinds of inhibitors: competitive, non-competitive, feedback inhibitors. Right? So that's about enzyme inhibitors. And next one, classification of enzymes. Classification of enzymes. So according to IUB system of classification, IUB, International Union of Biochemistry, International Union of Biochemistry. So according to that, what about a classification of enzymes? So here name of the enzyme and class of the enzyme generally based on type of the reaction catalyzed by that enzyme. That may be carboxylase, oxidase, concerned with carboxylation reaction, concerned with oxidation. So the nomenclature of enzymes maximum extent based on type of the reaction catalyzed by that enzyme. That may be oxidase, concerned with oxidation. So according to IUB system of classification, International Union of Biochemistry. Okay, so they classified enzymes into how many classes? According to that IUB system of classification, total number of classes number of classes of enzymes. 6 classes. So here criteria for the classification of all these enzymes into 6 was mainly on the basis of a type of reaction catalyzed by that enzyme. On the basis of a type of reaction catalyzed by concerned enzyme, they classified all these enzymes into 6 classes. So main criteria is type of the reaction. Type of a reaction. And each class with a certain number of subclasses. What about the total number of subclasses? 4 to 13 subclasses. Each class with 4 to 13 subclasses. And according to this IUB system of classification, generally each enzyme, according to classification of enzyme, that classification, so enzymes are with a 4-digit enzyme code. On the base of that, we can identify 4-digit enzyme code. First digit. Okay, so first digit. That represents class of that enzyme. Second digit, subclass of that enzyme. Third digit, subclass. Fourth digit, serial number of that enzyme in that class, concerned class. So first position, second one, third one, fourth one. How many digits? 4 digits, according to IUB system of classification. This is called as enzyme code, EC, enzyme code. Okay, so on the basis of that nomenclature and enzyme code, we can identify the concerned enzyme. And each enzyme ends with, what about the enzyme name? For example, some enzyme, whatever the enzyme, name of the enzyme plus suffix is ASE, age. So in the name of the enzyme, the suffix is ASE. Okay, so what are the 6 classes of enzymes? So first one, oxidoreductages. Oxidoreductages. They're also called as dehydrogenases. Dehydrogenases. So in the class name, what about the last word? Last one, ASE. The suffix is ASE. Second class. Second one, transferages. Transferages. Here also that ends with ASE. Transferages. Hydrolases. Next one, hydrolases. Fourth one, liases. Fifth one, isomerages. And the sixth one, ligases. So in all these names, class of names of enzymes, end with ASCH. Okay, so now what about the role of all these enzymes one by one? First one, oxidoreductages. So oxidoreductages means they are concerned with oxidation of substrates. So usually involving transfer of hydrogen atoms. Okay, so first one is oxidoreductages or dehydrogenases. Oxidoreductages. Okay, so these enzymes cause oxidation of one substrate and reduction of other substrate. First one, dummy reaction. So given one is a substrate that is in reduced form. So this one is in reduced condition. Simply I'm using R. That already in reduced condition, having lot of hydrogen atoms and electrons, for example, only. And the second S is nothing but substrate. The symbol, remember this symbol. This one is in oxidized condition, means able to accept reducing power. So now, for example only, here I'm taking 2H plus, for example only, 2H plus. So these enzymes are concerned with shifting of or transfer of hydrogen atoms or protons from this one to this one. So now what about the product? So first one, substrate, first one is in oxidized condition. Now plus S1, not one, that is a symbol only, hyphen better. Okay, so reduced. So according to given title, yes, these enzymes are concerned with oxidation and reduction. Oxidation of this one, reduction of this one. And that involves transfer of hydrogen atoms. This is the dummy reaction. In NCERT, only dummy reaction is given. What about the live reaction given in Telugu Academy textbook? So what about the given reaction? Malate, malic acid, malic acid plus one molecule of NAD plus. Okay, so that is a NAD, nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide. So this one is in reduced condition. This is in oxidized condition. Now take two hydrogen atoms, 2H plus and two electrons also. Two electrons also, but here given one is according to textbook, two protons, 2H plus given. Proton transfer of hydrogens given. You can take electrons also. So now, what about oxidized form of malate? OAA, oxaloacetic acid, oxidized form of that one. Plus, and what about NAD, reduced, NADH, H plus, reduced. So NAD is a coenzyme. We'll see what about this NAD at the time of cofactors. Just remember, NAD is nicotinamide adenine dinucleotide. Two nucleotides are present. Okay, so this reaction is present in TCA cycle, Krebs cycle, TCA cycle, Krebs cycle. That is part of a stage of aerobic respiration. So that is about oxidoreductages. And second class is transferases. So what about transferases? They are concerned with transfer of a group. Transfer of a group other than hydrogen atoms. What about the given example, dummy one, substrate bond, a group plus substrate without group gives rise. So this substrate is transferred to this substrate, a reactant. So first one without that group plus second one with that group. It's a dummy one, not the real one. So transferases, they involve transfer of groups between two substrates other than hydrogen atoms. Here, this group is not hydrogen atom. Some other, we'll see with the help of some example. The given example, glucose molecule, glucose plus ATP. Okay, so glucose, first one, ATP, first one, for example, first substrate, glucose, ATP. So gives rise weight. So from ATP molecule, one inorganic phosphate is added to glucose molecule. So now the product is a glucose 6-phosphate. Now glucose is with one phosphate molecule. Okay, so here in between these two molecules, what about the group shifted? That is a phosphate from ATP molecule. So ATP is a donor of that group. Inorganic phosphate is not, it is not hydrogen atom. Glucose 6-phosphate plus one molecule of ADP produced. Okay, right? And here, write one enzyme, malate dehydrogenase. Malate dehydrogenase. And here, hexokinase. Here I'm writing hexokinase. Hexokinase. Generally, kinases are concerned with a transfer of phosphate. So in this, according to this reaction, we can say transferases are phosphotransferases. Phospho or not? Phosphate molecule is phosphate molecule. Hexokinase. Phosphate molecule is transferred between these two. Okay, right? This is a hexokinase. Right? So that is about a second class. Right? Next, third one, hydrolases. Hydrolases. Hydrolases means hydrolysis. Hydrolysis of various bonds. Okay, so they're concerned with hydrolysis of a peptide bond, glycosidic bond, okay, ether bond, CC bonds. Such a number of examples given. Remember those. Hydrolysis of a ester bond, ether bond, CC bond, C halide bond. Here I'm giving some examples. Hydrolysis of a, they're concerned with hydrolysis of ester bonds, ester, ether, peptide, CC bonds, C halide, like that. So many examples are there. Peptide bond also. So here it involves hydrolysis means removal of water. Removal of water, but not hydrogen atoms. Removal of water. So they're generally concerned with dehydration generally. Hydrolysis means breaking of ester, ether, peptide is nothing but dehydration generally. Hydrolases. 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 So what about the given example? In this case, the given example is fructose 1 comma fructose 1 comma fructose 1 comma 6 bisphosphate. This one is fructose. Come back, glucose 6 phosphate. Sixth carbon received with inorganic phosphate. Come back. This one is fructose. F is nothing but fructose. 6 carbon sugar. Fructose 1 comma 6. How many phosphates are there? 2 phosphates. Biphosphate. Fructose 1 comma 6. First carbon with 1 inorganic phosphate. Sixth carbon with 1 more phosphate. Total number of phosphates 2. So it is a fructose 1 comma 6 bisphosphate plus, in this case, addition of water. By adding water, they can break. Here breaking, actually addition of water. In this case, addition of water. Take addition of water. Actually, ester ether peptide bonds are formed by dehydration, and these are concerned with the hydration, addition of water molecules. By adding water molecule, they are able to cause hydrolysis. So addition of water, go for addition of water. Don't go for dehydration, sorry. Addition of water. Hydration reaction, sorry. Hydrolysis, they are able to add water molecule with the help of which they are able to break bonds. So fructose 1 comma 6 bisphosphate plus 1 H2O molecule gives rise fructose 6 phosphate plus 1 inorganic phosphate. 1 inorganic phosphate is eliminated from first one, that is a biphosphate. So fructose 1 comma 6 bisphosphate is changed into fructose 6 phosphate. 1 inorganic phosphate is eliminated. Enzyme required is a bisphosphatase. Bisphosphatase. Fructose 1 comma 6 bisphosphatase. So hydrolysis are able to cause hydrolysis of bonds by the addition of water molecule. Addition. Okay. Fourth class, liases. Liases. Okay, so liases are concerned with splitting of substrate or removal of groups from a substrate other than hydrogen atom and leaving double bonds. What is liase? Simply splitting of substrate and removal of a group and leaving double bonds. First dummy reaction, carbon bond carbon, bond X. This is a dummy one. Bond Y. So now all are with single bonds only. So liases are concerned with separation or splitting of a substrate and leaving double bonds. So between X and Y, no bond. Now X bond Y plus C basically with CC with the one bond only. Now 2 bonds. Double bond. That is dummy reaction. So what about liases? They involve removal of a group from substrate and development of double bonds. Okay, so now here I'm taking one example, live example, arginosuccinic acid. So that is one organic acid, arginosuccinic acid. So what about the fate of this arginosuccinic acid? Is cleaved into arginine plus fumaric acid. So 2 products are produced. Okay, so arginosuccinic acid is cleaved into 2 products. What are they? Arginine and fumaric acid. Okay, so what about the definition for liases? They are able to remove groups from a substrate and leaving double bonds. according to dummy one, C bond, single bond, C, and to which X and Y are connected with a single bond only. So development of a single bond between X and Y, and development of a double bond between C and C, which are having single bond only initially. So that's about liages. Now, fifth one. Fifth one, isomerages. Fifth class, isomerages. Okay, so they are concerned with changing of one isomer into other isomer. So what are the isomers and isomerization? Okay, so geometrical isomer, okay, positional isomer, okay, positional isomer, geometrical isomer, okay, optical isomer. So from one isomer to other isomer. Discuss with your chemistry sir. What is a geometrical isomerization, positional, optical? Actually, you have to study a number of kinds of isomerizations in chemistry. Use that knowledge and discuss with your chemistry sir. Geometrical isomer into positional, positional into optical, and vice versa. Okay, so here the given example is a ribose 5-phosphate. It is one type of sugar, 5-carbon sugar, ribose 5-phosphate. So is isomerized into ribulose 5-phosphate. Okay, ribulose 5-phosphate in the presence of a ribose 5-phosphate isomerase. Okay, so this reaction is present in C3 cycle. We will see this one later. C3 cycle reaction. And the last one, sixth class, is ligases. Okay, so ligation, attachment, DNA ligase, like that. DNA ligase, RNA ligase. Go back to molecular base of enzymes, you'll get it. Ligases are concerned with the attachment of two compounds, linking of two compounds. Here, ATP is consumed. So ligation means attachment, linking of two substrates. Okay, so what about the given reaction? Glutamic acid. Acidic amino acid. Is amino acid. Glutamic acid, amino acid, plus one molecule of NH3, ammonia, plus one molecule of ATP. Okay, so glutamic acid plus amino group. Already this is amino acid with amino group 1. And the second amino group is going to join with glutamic acid. So gives rise glutamine. So glutamine is amide. Go back to mineral nutrition, amide plus ADP plus inorganic phosphate. So during this reaction, one molecule of ATP is consumed. And enzyme required is glutamine synthetase. Glutamine synthetase. Okay, so ligases are concerned with linking of CO, C bond O, C bond S. Okay, so they are able to link CS, CO, PO bonds also, phosphate and oxygen, CN bonds. Okay, so that's about classification of enzymes. Now, enzyme code. Enzyme code, EC. So one example is given in our textbook, that is, the given example is enzyme code of glucose 6-phospho transferase. Okay, so old name of this one, hexokinase. Okay, so what about EC of this one? Enzyme code of this one is 2712. 2712 enzyme code. So 2 means second class. So what about second class? The second class is transferases. Second class, subclass, seventh subclass. Next, first subclass, serial number of that enzyme in that class. Okay, so if 1 is given, first class. If 2 is given, second. Third is 3 is given, third class, like that. So on the base of first digit, we can identify the class name to which concerned enzyme belongs. Okay, so here in this place, first digit is either 1 or 2 or 3 or 4 or 5 or 6 only. Okay, and this one subclass, this one subclass, and the serial number. So 2712 is enzyme code of that one. First one, first digit, class name. Second one, subclass. Third one, subclass. And this one, serial number of that enzyme in that class. So how many digits? 1, 2, 3, 4. 4-digit enzyme code. And the base, no, wait. So that is about enzyme code. So that is about classification of enzymes and enzyme code.